Hi everyone, welcome to the second part of our discussion on laboratory hazards and safety. And as you remember last time, we were able to discuss about laboratory hazards, what are the different hazards that medical technologists are exposed to inside the laboratory. So this time, we will be discussing about laboratory safety, or what are the things that we need to remember or do in order to avoid the hazards that we are exposed to inside the lab. We have the chain of infection. The chain of infection is composed of three main components that are linked together. In order for an infection to occur, there must always be the presence of three components. Our goal is to always break the chain of infection. That is to prevent the spread or occurrence of a disease or illness. This is the chain of infection. As you can see, there are three components in the chain of infection. First, the susceptible host, usually human or animals. We also have the source, which is usually an infectious agent, and the mode of transmission. In each of these three components, we can find the different ways on how to destroy or mitigate the component to prevent disease from happening. For example, to destroy the infectious agent or the source, we need to perform proper hand washing, correct disposal of biohazard waste as well as specimen bagging. We also need to perform the contamination or disinfection of our working area. Next, to prevent the host from acquiring the infectious agent, he or she must follow standard precautions, receive immunization, practice a healthy lifestyle, perform exposure control plan, and for some instances that he or she is exposed, should receive post-exposure prophylaxis. Lastly, to prevent the transfer of the infectious agent to the host, hand washing must be done. The person must be in complete PPE, especially if he or she is processing specimens inside the lab, performing practices that do not lead to aerosol formation, using of sterile or disposable equipment, and pest control plan, since diseases can be also transmitted through vectors like mosquitoes, flies, ticks, and other pests. Aside from the three components I have mentioned earlier, such as your host, your source, and the mode of transmission, we have additional three components in the chain of infection. These are the portals of entry, the portals of exit, and the reservoir. When we say portal of entry and portal of exit, these are parts of the human body where microorganisms can enter and start to infect our system. So in order to break that component, for example, in the portal of entry, the person must practice hand hygiene, follow standard precautions, um, wear complete personal protective equipment, and use sterile equipment. The same goes with your portal of exit. In order to break the link, we have to seal our biohazardous waste container properly. We also have to properly seal our specimen containers because that could be also a source of infectious agent. Practice hand hygiene and follow standard precaution. And lastly, for the last component of the chain of infection, which is the reservoir could either be humans, animals, insects, fomites, and also our blood or body fluids or the specimens of our patients. So in order to break this link, in order to break this component, we have to perform proper disinfection and also practice hand hygiene. We now go to laboratory safety. So we have here the definition of our laboratory safety. It says that lab safety is a careful process with the goal of preventing accidents and injuries among students and also laboratory staff. Laboratory safety is only effective when rules are strictly imposed and when individuals follow these rules properly. If you take a look at the illustration above our definition, we can see people working inside the laboratory, and we can see here some descriptions that will tell us if that person is properly following the protocols set inside the laboratory. So take for example this person here. He is trying to experiment on himself by placing the sample on his eyes, which is very incorrect because we do not know the nature of the samples we are processing inside the lab. So we do not let these samples touch or get inside our system through our mucous membranes. Okay, remember our universal precaution that we need to treat all laboratory specimens as though they are potentially infectious. We also have here another person. He is tasting the chemical he is experimenting on. This is also very wrong because we do not know the nature of the chemical. It could be corrosive. So once it comes in contact with your tongue might disappear. Okay, then we also have do not eat or drink inside the laboratory. This is a very important precaution inside the laboratory that we do not allow students to eat inside the laboratory. Okay, if you want to eat, you can do it outside or in the pantry. And then we also have know what to do in case of an accident. If you can still remember what we discussed about our fire extinguishers, if there are accidental fires inside the laboratory, always remember the acronym PASS and RACE. Okay, and then we have dispose of waste properly. So you need to learn how to categorize the waste you are generating inside the laboratory and learn how to dispose them properly. For example, if your waste is a sharps, you need to dispose it or throw it off in its designated container, or if the waste is an infectious waste, it's placed inside a waste container with a yellow um, bag. Okay, so that's designated for your infectious waste. For laboratory safety and regulations, we always need to take the time to be knowledgeable about lab safety concerns and practice proper safety procedures. This is for our own good and also to ensure that accidents can be prevented from happening. In most cases, safety is a combination of common sense, good behavior, and housekeeping. If these three are present, then everything will work smoothly inside the lab, and no harm shall come to any laboratory staff. The lab must be systematic in its operations and safety protocols all the time. These are the general laboratory safety guidelines that a laboratory staff should follow when working inside the laboratory. So first, we have conduct yourself in a responsible manner at all times and wear appropriate PPE. So again, you're not allowed to process patient specimens if you're not wearing your complete personal protective equipment. Number two, do the laboratory activity in the presence of your instructor and follow instructions, especially if you do not know the procedure very well. It is required that you be trained under the supervision of a head medical technologist because they will guide you on what to do or what not to do when working in the section. Number three, study and familiarize the lab procedures before any laboratory activity. So if you are unfamiliar with the procedure, this is your chance to refresh your memory by reading what the procedure is all about, what's the principle behind the procedure, so that by the time that you're about to perform the activity or the procedure, it would be very easy for you. Number four, perform only those assigned laboratory activities. So you are not allowed to perform anything beyond what is assigned of you to do or the activity for that day. You're not allowed to perform anything unless specified by your head medical technologist. Number five, eating, drinking, chewing gum, and applying makeup or lipstick are prohibited during the class or during the activity. Okay, so this is self-explanatory. Number six, clean your working area before and after any laboratory activity. This is a protocol every time there are laboratory procedures to be done. Before you start working and after you work or process your specimen, you really need to clean your area. Number seven, if by any chance there is broken glassware, wrap it in a box and label it before disposing. So remember that our broken glasswares, they are considered as sharps hazard, so you have to dispose them properly. Number 8, avoid mouth pipetting or using your mouth to suction any chemicals or agents. Okay, so today guys, we are very lucky because we are born in a generation that do not perform mouth pipetting of chemicals. We already have our serological pipettes. We also have automatic pipettes. So please, we don't need to pipette our chemicals using our mouth. Okay. Number 9, if there is a spill during the activity, clean up immediately. And number 10, notify the instructor any problems or accidents that may have happened during the activity. So this is also very important that you need to address the situation immediately. And one way of doing that is to inform your teacher or your head medical technologist that an accident happened so that it can be properly documented. Again, we have Occupational Safety and Health Act or OSHA. This is a federal law that mandates safe working conditions for workers, even laboratory technicians. So, um, personals or people working with OSHA may inspect workplaces for compliance with safety rules, and compliance is the law. It's not just a nice idea. So what are the mandatory laboratory practices being monitored by OSHA? We have the following: bloodborne pathogens, we have the use of your formaldehyde, and then we also have respiratory standards and the wearing of personal protective equipment. Under Occupational Safety and Health Act, we also have individuals who are categorized into three under exposure control. So we have category 1 if a person has a daily exposure to blood and body fluids, category 2 for a person with regular exposure, and category 3 with no exposure to blood and body fluids. So employers of a certain laboratory must offer hepatitis B vaccine for free to all personnel in categories 1 and 2 since they are always exposed to specimens that, with visible blood or specimens that are body fluids in nature. Other regulations and guidelines that we need to know about, we have the Clean Water Act. This is a federal law that regulates the discharge of pollutants into the nation's surface water. That includes our lakes, rivers, streams, wetlands, and coastal areas. Because we all know there are also other big companies that dispose their chemicals into large bodies of water, and these chemicals not only pose a hazard to the people living near these bodies of water but also to the environment as a whole. Then we also have the Toxic Substances Control Act or TSCA. It's a law that regulates the introduction of new or already existing chemicals to evaluate, assess, mitigate, and control risks which may be posed by their manufacture, processing, and use. So again, many chemicals we use inside the laboratory, when they are not properly handled or disposed, they may cause harm to aquatic organisms or the environment. So this law tries to protect our environment by monitoring the use and the disposal of the chemicals that we are using. And then lastly, we have the Joint Commission on Accreditation of Healthcare Organization. JCAHO is an accrediting non-profit organization that developed the idea of having a systematic standardization in hospitals. So standardization is very important because um, through standardization, we can produce quality results of the laboratory tests that we are performing inside the laboratory so that we are at par with the other healthcare organizations or laboratories worldwide. Also, if you are working in a JCAHO accredited laboratory, it would be very easy for you to find work abroad. Okay, because yes. For safety awareness, we have two key players here. The first one is the employer, who provides and supervises safety methods, equipment, and policies, and the employee, who understands and practices established safety procedures. The employee also notifies supervisors of unsafe conditions. So these two must work together in order to ensure that everything that is being done inside the laboratory is safe. That's why the bottom line is safety is everyone's responsibility. It's not only the responsibility of the employer, but it's also the responsibility of the employee. So both of these key players, again, should work hand in hand. For labeling, of course, everything must be labeled. Hazardous material must include all necessary safety information, and we have the appearance of our diamond-shaped color-coded NFPA symbol. It indicates the nature of the chemical inside the container. By looking at your NFPA symbol, it will give you an idea of the information on the flammability, the stability. We also have the specific and health hazard related to the chemical inside the container. So in this corner here, we can see a photo of improperly labeled chemicals. These are very dangerous chemicals when placed inside a laboratory because they don't have any names, and that's why if they don't have any names, we do not know the nature of this chemical. It may be flammable, it may be corrosive, it can be an irritant. So unlabeled containers inside the laboratory, it's a no-no. You really have to place a label and also indicate the NFPA hazard symbol in your container. Okay, that's why labeling is very important because it gives you an idea of how to handle the chemical in the container. For chemical safety inside the laboratory, the following protocols must be set by the laboratory to ensure that all people who are handling chemicals are safe from the hazards brought about by these reagents. So first, we have the hazard communication. The lab must provide written policies for the safe use of hazardous chemicals. This is also part of OSHA regulation. So if your laboratory is OSHA regulated, it must adhere to the rules and protocols set by OSHA when it comes to chemical safety. Next, we have Material Safety Data Sheet, or the MSDS. This lists all the safety information created by the manufacturer for each laboratory agent, may it be biological or chemical. And our laboratories, they are responsible for gathering all the MSDS information available for all our agents and chemicals. These MSDS information must be easily accessible to any laboratory staff, just in case accidents may happen concerning to the reagent. And then employees must have ready access to MSDS information. The third here is your laboratory standard. Laboratories must have written chemical hygiene plan that provides specific work practices for hazardous chemicals. So, in cases of accidental chemical spills, what must be done, what should the laboratory technologies do in order to avoid being exposed to chemical spills. So, all of these are contained in your chemical hygiene plan. And lastly, we have the storage and handling of chemicals. Next, we have the chemical hygiene plan. So, OSHA requires all facilities that make use of hazardous chemicals to have a written chemical hygiene plan available for all employees. The chemical hygiene plan must contain the following. Number one, we have appropriate work practices. Number two, standard operating procedures. Number three, the proper wearing of personal protective equipment. Number four, engineering controls such as fume hoods and flammable safety cabinet, employee training requirements, and lastly, medical consultation guidelines. So basically, the contents of the chemical hygiene plan, it details the different steps that will guide the medical technologists on what to do on especially in handling chemicals that are being used inside the laboratory. So what are the things that we need to remember to ensure our safety, especially in handling chemicals? So we have the following. Number one, use fume hoods whenever possible in dispensing or pipetting dangerous chemicals. For chemicals that emit large amounts of fumes, fume hoods are very useful because they prevent us from smelling the vapors emitted by our dangerous chemicals. Number two, safety goggles, glasses, face shield, and lab gown should be worn at all times in the laboratory, and remove it when leaving the laboratory. So this is a very common mistake for some because they tend to wear their laboratory gowns even outside the lab room. Okay, when walking in the hallways, when walking on the streets, you really need to remove your laboratory gowns and before going out. That's the protocol there. Number three, gloves should be worn when handling blood and other body fluids and should be disposed to the appropriate trash bin. So usually for soiled gloves, you have to dispose them in infectious waste. So that is your waste container with a yellow lining. Also, when your gloves are already torn, you have to replace them. You have to replace them with new gloves. And another thing that we need to remember is before we wear our gloves, we need to check first our hands if we have visible wounds, and we have to cover our wounds first with band-aids before we wear our gloves. Number four, wash hands before donning gloves and after doffing of gloves. So of course, hand washing, it's one way of breaking the chain of infection. It's the most effective way of breaking the chain of infection. Number five, tie long curly hairs and remove any jewelry including your watch. If you are wearing bracelets that have dangling parts, you need to remove your bracelet. So you can just wear them after you perform your laboratory procedure. Number six, do not apply cosmetics in the laboratory. And number seven, eating, drinking, and using cellular phones are not allowed in the laboratory. So those are the things that we need to remember in order to ensure our safety in the inside the laboratory and also in handling chemicals. So how about if there are chemical spills inside the lab? For this, we can make use of our chemical spill cleanup kit. So these are the following procedures that we need to follow to clean up accidental spills or chemical spills inside the lab. First is we wear our appropriate PPE when cleaning up spills. Remember, our PPE is composed of our lab gown, our gloves, and our mask. And we also have goggles or face shields. Number two, we need to know if the accidental chemical spill is an acid spill or a solvent spill. So if it's an acid spill, we have to apply a neutralizer, usually in the form of sodium bicarbonate, to the perimeter of the spill, and then we have to mix thoroughly the spill with the neutralizer until fizzing and evolution of gas ceases. So the third step is to transfer the mixture to a plastic bag, tie shut, fill out a waste label, and place inside the fume hood. Then what if our spill is a solvent spill? So these are the steps. Apply activated charcoal to the perimeter of the spill. Mix thoroughly until material is dry and no evidence of solvent remains. And lastly, transfer the absorbed solvent to a plastic bag, tie shut, and place inside the fume hood. So again, there's a difference between cleaning up acid spills and solvent spills, because your acid spills, you need to make use of a neutralizer in the form of your sodium bicarbonate, and your solvent spills, you need to apply your activated charcoal. For storage of chemicals, these are the following steps that we need to remember. Letter A, flammable areas should be cool enough to prevent ignition when the vapor mixes with air. Adequate ventilation should be provided to prevent vapor buildup. So it's not enough that you have to store all your chemicals in a very enclosed area. You also have to make sure that the area for the chemical storage has a proper ventilation. Letter B, flammable materials should never be stored near acids because this can have a violent reaction which may lead to explosion of the chemicals. Letter C, avoid storing flammable materials in conventional refrigerators. Sparks generated by internal lights or thermostats may ignite flammable material inside the refrigerator, causing an extremely dangerous explosion hazard. So remember guys that the refrigerators that we use at home, they are different from the refrigerators that are seen inside the laboratory. So if your laboratory wants to purchase refrigerators, you have to make sure that those are the refrigerators um, that are appropriate for laboratory use. When handling of chemicals, we need to remember these following things. First, use gloves and safety goggles when handling flammable liquids or vapors, since some of the chemicals emit fumes that are very irritating to our eyes and nose. So using of safety goggles is really recommended. Letter B, dispensing of flammable or combustible liquids should only be done inside a fume hood or in an approved storage room. Okay, for chemicals that emit toxic fumes, please do not process or deal with them in an open working area. You have to place them inside chemical fume hoods. And number three, do not use water to clean up flammable liquid spills. To prevent fire from happening inside the laboratory, these are the things that we need to remember. Number one, be aware of the ignition sources in the lab area, that such as your electrical equipment, heat sources, and sockets. Number two, never store flammable liquids in the standard refrigerators. I think we have mentioned this in the previous slide. Explosion-proof refrigerators are needed for storage of flammable liquids. Number three, store flammable liquids in appropriate safety cabinets or safety cans. And number four, make sure the electrical cords are in good condition. Always check for destroyed insulators of your appliances. All electrical outlets should be grounded and should accommodate a three-prong plug. Remember our electrical hazards that we should not overload our electrical sockets with plugs, and we have to check for presence of chipped or destroyed electrical insulators to prevent us from experiencing electrocution. For waste collection in the laboratory, these are the following things that we need to remember. Number one, dispose all biohazardous substances in a biohazard bag. Usually in the laboratory, all infectious wastes or pathological wastes are placed inside a yellow bag or a yellow waste receptacle. Number two, dispose swab wrappings, band-aid wrappings, use paper towels, kit boxes, and any other non-biohazardous waste in regular trash bags. Usually, the color of the trash bag for this one is the black colored container for non-infectious waste. Number three, dispose all use needles, pipettes, broken glass, and slides in plastic biohazardous sharps container. Usually, it's a red colored waste container for the sharps. Number four, dispose all used tubes in red biohazardous bags. Number five, dispose all bacterial and fungal culture plates in yellow biohazardous bags, because our bacterial and fungal culture plates, they are pathological waste or infectious waste. They have the capacity to transmit or cause infection to other humans or animals. And number six, do not throw non-biohazardous waste into the red or yellow biohazard bags. Always remember our non-biohazardous waste that are dry are disposed in black colored containers, whereas non-infectious wet waste are placed inside green colored waste containers. Next, we have our personal protective equipment or PPE. So PPE, it's a very important part of being a medical technologist, because you need to wear this in order for you to be protected from the infection brought about by the specimens we process inside the laboratory. Our personal protective equipment, it's composed of your gloves, your fluid-resistant lab gowns, your goggles, masks, and face shields. So you have here that your gloves should be worn when in contact with patients, specimens, and laboratory equipment or fixtures. When specimens are collected, gloves must be changed between every patient. You can also change your gloves when you think that it's already soiled or very dirty, or when it's already torn. So you have to change uh, your gloves. You have to throw them away in infectious containers, and you have to have a spare for you anytime you need your change of gloves. Next, we have fluid-resistant laboratory gowns with cuffs. It must be worn completely buttoned, and gloves should be pulled over the cuffs. Okay, and then the third one is our face shields, our masks, and goggles. These materials, they protect our mucous membrane of the eyes, nose, and mouth from specimen splashes and aerosols. So again, if you do not have your personal protective equipment, you cannot process any specimen inside the laboratory. Then we have hand washing. Hand washing, again, it's the most effective way of breaking the chain of infection. When done correctly, it's the single most effective way to prevent the spread of communicable diseases in all settings. So when we perform hand washing, we need to sing a song, and this song is called Happy Birthday. Happy Birthday is a hand washing song. So when you perform hand washing, you need to sing Happy Birthday twice in order to perform a very effective hand washing. Now we go to laboratory safety equipment. These are the materials we see in the laboratory that, although they look normal or ordinary, they are so useful in times of need, or simply they protect us from hazards inside the laboratory. We start off with our individual storage containers. Choosing the right storage container depends on the reagent's compatibility with the container. A safety can is an approved container of no more than 5-gallon capacity. It has a spring closing lid and spouts cover and is designed to safely relieve pressure buildup within the container. So again, our individual storage containers, they must be properly labeled with the name of the chemical and also the hazard associated with that chemical and the NFPA color-coded symbol to give us an idea of what is the nature of the chemicals stored in that container. Next, we have our laboratory chemical fume hood. It has the ability to capture, contain, and expel emissions generated by hazardous chemicals. We have mentioned earlier that some chemicals have the ability to emit toxic fumes. That's why they must be placed or handled inside the laboratory chemical fume hood. This is a type of a local ventilation device designed to limit the exposure to hazardous or toxic fumes and vapors. So, this is also one way of protecting the person handling the chemical because there is a barrier in the form of the lid that separates the chemical from the person handling the chemical. So, if the chemical emits fumes or vapors, the person handling the chemical would be prevented from inhaling the chemical directly because it's processed inside the chemical fume hood. Then we have our laboratory refrigerators. Again, the refrigerators we have inside the laboratory, they are different from the refrigerators we have at home because these refrigerators are designed to store chemicals, to store reagents that are used when processing patient specimens. So refrigerators, they are not appropriate for storing flammable substances, and they are usually equipped with temperature monitoring device to warn the medical technologists that the refrigerator might not be in the correct temperature. Temperature monitoring is very important for some reagents inside laboratory because it maintains their viability when stored inside the refrigerator. Refrigerators used for food and beverage storage should be labeled. Each refrigerator and freezer must be monitored daily to ensure proper functioning. And um, label is very important. You have to place it on your refrigerator that no food or beverage may be stored in this refrigerator or freezer. So if you want to store food inside the laboratory, you can purchase a separate refrigerator solely for storing your food and beverages, and you don't need to put that in any of the main sections of the laboratory, but you have to place the refrigerator in the pantry. Then, containers placed in the refrigerator must also be completely sealed or capped by using paraffin or cap, labeled and securely placed inside the refrigerator or freezer. So, to avoid spilling of um, solutions placed inside certain containers, you have to cover them properly or seal them properly. Then we have our chemical storage cabinets. Our chemical storage cabinets are used to store flammable and corrosive chemicals inside the lab. These cabinets should be well ventilated, and storage of these chemicals should be limited to small quantity as possible. And also, we don't want our chemicals to be stored together with other chemicals that may cause explosion. So we have to properly group our chemicals based on their characteristics and qualities to avoid these types of accidents. Then we have our safety showers for accidental chemical spills to the body. Our safety showers provide an effective means of treatment if chemicals are spilled or splashed onto the skin or clothing. You don't need to take off your clothes when there are accidental spills. If that happens, you immediately run to the safety shower and pull this lever or this handle to let a strong gush of water flow out of the shower. Okay, so again, please do not take off your clothes. You run immediately under the shower and pull the lever. That is the proper use of our safety showers. So we also have the eye wash station. So the eye wash station, it's a unit for washing chemicals or substances that might splash into an individual's eyes before he or she can seek further medical attention. So for accidental chemical spills to the eyes, we have this laboratory equipment. This is our eye wash station. So you just have to position your head near this portion here and push this lever or handle. So a strong force of water will come out from this area and will hit directly your eyes or the part of your face that is affected by the accidental chemical spill. So always remember that if that happens, you need to flush the area first for 15 minutes with running water before you seek medical attention. So it's not the other way around, wherein you need to go first to the emergency room before you wash your eyes with water. Our eye wash station, it serves as a first aid for accidental spill. So again, you need to flush the area with water first for 15 minutes before you go to the hospital to seek medical attention. Then we have our spill kits. A spill kit, it's a collection of items to be used in case of a spill, leak, or other discharge of oil. They are developed so that a prompt response and cleanup may be performed. So every person working in the laboratory should be trained or have an idea on how to handle spills inside the laboratory. It usually contains 2 or 3 types of equipment, such as personal protective equipment, equipment used to clean up the spill, or equipment to stop leakage. Then we have our biosafety cabinets. Guys, our biosafety cabinets, it's different from the chemical fume hood. Okay, our biosafety cabinets, it's also known as our microbiological safety cabinet, which is an enclosed ventilated laboratory workspace for safely working with materials contaminated with pathogen requiring a defined biosafety level. So don't worry, because there is a separate lesson for the biosafety cabinets, because biosafety cabinets, it has four types or four levels that we need to know, and our microorganisms will be processed depending on the kind of biosafety level it requires. Okay, so that's the purpose of our biosafety cabinets. You can see biosafety cabinets in the microbiology section when we culture our microorganisms in culture media, when we grow microorganisms from our urine or stool samples, we make use of our biosafety cabinets. Then we have our fire alarms. Our fire alarms are designed to alert endangered personnel if in case of fire. If you can still remember the acronym RACE, which is used to when accidental fires are discovered. A there is alarm, which means to activate the institutional fire alarm to alert all people in a unit or building that a fire is happening so that they can be evacuated to safety. Okay, so most fire alarms are in the hallway and usually inside a red box with a label fire alarm. We also have our fire extinguisher. This is also one of our laboratory safety equipment. This helps us put off or extinguish fires inside the laboratory. So please do not forget the acronym that we discuss or use when operating a fire extinguisher. We have the PASS. And also, please do not forget the different classes of fire, the types of fire, and the fire extinguisher that is required to put off a certain type of fire. So please do not forget that, that's very important. Then we have our fire blankets. It says here that all laboratory personnel are discouraged to use fire safety blankets to extinguish the fire. Fire safety blankets should be used to keep shock victims warm. Okay, so that's the purpose of our fire blanket. It's not used to put off fire inside the laboratory, because we have our fire extinguisher. So the main purpose of using the fire blanket is to give comfort to our fire shock victims. Lastly, we have our biohazardous waste and sharps container. So these are our sharps container, the red colored one and the yellow colored sharps container. And maybe you have seen these containers inside the lab when you had your blood collected for the hepatitis test. Okay, so um we usually use the sharps container in disposing our used needles after collecting samples from our patients. So we have here the characteristics of a good sharps container. Number one, it must be puncture proof, which means when you discard your sharps inside the container, it does not create a hole on the wall of the container. Okay, next, it must be properly labeled. What are the things that we need to see, or so that we can know that our sharps container is properly labeled? First, we have it must be labeled with sharps. And number two, you must be able to see a biohazard symbol, because that should indicate that all the waste being thrown in this container is considered infectious. Next, it must have a cover to prevent spillage of contents. When your sharps container is already full, you need to close the lid, and then you need to get another empty sharps container to dispose your used needles. Next, it must be ideally should not be made of fragile material. Okay, most of the time, our sharps container are made of plastic, but again, the container must be puncture proof. And it may have grooves to facilitate ease of needle removal. In some of the sharps container, after collecting your sample, you need to just place the needle of the syringe at a certain portion of your sharps container, and then you just have to rotate your syringe and automatically the needle drops off inside your sharps container. So that's the purpose of the groove there. Because in some hospitals, they manually remove the needle from the barrel by using the forceps. But it's because their sharps container cannot provide a groove for them to insert their sharps and twist the barrel so that the needle will just have to fall off inside the container. So, yes. Again, safety begins with recognition of hazards and is achieved through the application of the following. You have common sense, a safety-focused behavior or habits, good housekeeping, and a continual practice of good laboratory technique. So these are the following characteristics that a professional medical technology should embody or should show inside the laboratory to prevent accidents from happening. So always remember that you need to always take time to be safe. So thank you for coming to our discussion.